Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from neuroscientists across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of our members here. Each seminar focuses on one of the new interdisciplinary themes of Cambridge Neuroscience, which we will be launching later this year. For more info on the talks covered in this seminar series and all things neuro related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below.
So today I'm very delighted to welcome you all back to the Easter term of the Cambridge Neuroscience Interdisciplinary Seminar Series. I was just saying to everyone just before we pressed record that we have a great lineup of speakers for you this term with Alan Harbison, Madeleine Lancaster, James Rowe and Simon Baron-Cohen. And of course, we're delighted to kick off the series this term with Professor Jeff Dalley. Jeff is a professor in the departments of psychology and psychiatry here at the University of Cambridge. I'm very lucky to work with him on the MPhil in basic and translational neuroscience, for which he is one of the co-directors together with Ole Poulsen. He's also a fellow of St. Catherine's College, and his work is broadly interested in behavioural and cognitive neuroscience, with a special interest in psychopharmacology and the real kind of translation of basic advances in neuroscience to clinical psychopharmacology in many different diseases. So with that, I'm going to pass you over to Jeff and I'm going to stop talking.
Okay, thank you, Dervila. That's very, very kind introduction. I guess after 2 years of online teaching and lecturing, I'm kind of prepared to go back to live speaking, actually, but perhaps I'm in the minority, I'm not sure. Anyway, today's session, I'm going to talk about one of our projects looking at early life stress, early life adversity, and impacts on the developing nervous system. And of course, in infancy and early life, stress has quite dramatic and persistent effects on the developing brain. And there can be many sources of stress, and I'm thinking in particular of uncontrollable stresses, for example, poverty, maternal deprivation, perhaps loss of a parent or both parents, trauma, abuse. All of these can be thought of as uncontrollable stresses, and they can have, as I say, dramatic effects on cognitive and emotional development. And perhaps as an extreme example of neglect comes from Romanian orphans. This was in the rather brutal and repressive government of Nicolae Ceausescu at Romania, where contraception and abortion was banned. And the idea here was that this would increase the birth rate and increase economic growth and prosperity. In reality, what happened was there was children who were abandoned and sent into these orphans. Some orphans were better than others, some were particularly grim, and it led to institutionalized neglect. And this had quite dramatic effects on the outcomes of these children. And as a collection, one can think of early life adversity, including abuse, neglect, exposure to violence, family circumstances, leading collectively to what we call toxic stress. Now, toxic stress is a form of stress which overwhelms an individual and our adaptive ability to cope. And of particular interest was an exploratory brain imaging study carried out by the Neuroimaging Centre at King's College London. And in this small cross-sectional study using structural volumetric MRI, control subjects as well as Romanian adoptees, these are adoptees taken into UK families, were scanned, and various white matter, grey matter segmentations were carried out, focusing on the corpus callosum, hippocampus, amygdala. Hippocampus and amygdala are particularly sensitive to early life stress. And the major outcome from this small sample study was an enlargement of the amygdala, particularly the left amygdala, but also the right amygdala as well. Now, this was a particularly important study to demonstrate that severe early institutional deprivation can have quite profound effects on structures, in particular structures in the temporal lobe, including the amygdala. One of the major serial causal connections with early life stress is with anxiety and depression. And there are two major ideas out there. The first idea or theory is about stress amplification, that if you're exposed to high levels of early life adversity, this somehow sensitizes you to later impacts of stress. So if you follow this top graph here, you can see whether the later stress is of a low intensity or a high intensity, that the effects of that stress is much greater in individuals with a history of early life adversity. There's another idea out there, and this is a related idea, that some stress early on is good for us. It inoculates and builds immunized stress effects. In other words, some stress can attenuate the effects of later stress. And presumably, the balance between amplification and inoculation must depend on the intensity, the duration of the stressor, in particular the controllability of the stressor. And there are modulators or moderators of these two theories, in particular genetic dispositions, which I'll talk about more in this talk, the impact or the nature of the later life stress, parenting, behavior, social support structures, coping mechanisms. The effects we see on depression are strongly sexually dimorphic, and all of these impacts ultimately will affect the signaling through the hypothalamic pituitary axis, through the stress hormone cortisol, as well as other stress hormones, for example, noradrenaline. These affect brain circuits, brain networks, and plasticity, and in extreme cases can lead to these neuropsychiatric disorders later in life. One idea which my group have been very interested in is this idea, and it's not a new idea at all, about multiple hits that individuals absorb through the life course, from early life stress, understanding single, dual, and poly-hit stress episodes. This is particularly relevant in infancy, early life, where synapses are being formed, grey matter density is expanding. From this image here in the lower left, you see from the 5 years of age, how the density of grey matter increases substantially, particularly in temporal frontal cortical regions. Post-childhood and then into adolescence, you can see how these synapses are pruned and synapses are lost, forming the adult phenotypes. What we know about this early childhood period is that there's a wide window for stress-induced plasticity, particularly through epigenetic modification. And it goes to stand that any stress, particularly uncontrollable or toxic stress during this period, can change the trajectory of developing neurons in the nervous system. One idea that we've also been working on quite closely is the idea that the stress impacts are working or hijacking the immune systems. And we know, even through an evolutionary perspective, that the stress and the immune systems are closely interconnected. And there have been many reports showing that prenatal early life stress has effects on microglial activation in the brain, that second hits and subsequent stress hits can prime these microglia to accentuate the effects of cytokine release, this toxic exhaust of chemicals into the nervous system, leading to their outcomes on depression, anxiety, and so on. The late developing frontal cortical regions, including the rostral anterior cingulate cortex, medial prefrontal cortex, subgenual anterior cingulate cortex, are particularly vulnerable to early stress impacts. And I've told you already that hippocampus and the amygdala are also strongly implicated as regions of that develop either atrophy in the case of hippocampus or hypertrophy in the case of the amygdala, some of these early stress impacts. So what's the mechanism which links stress with the immune activation? Well, here's one idea from Weber, published in 2017. Stress, of course, activates the hypothalamic pituitary axis, as well as the sympathetic nervous system. There are two routes whereby the immune systems can be activated. One is a central route, where there is local immune activation through the stress episode, activating microglia to release pro-inflammatory cytokines, for example, the family of interleukins, as well as chemokines. The chemokine substances have a secondary effect to recruit monocytes from the peripheral circulatory system. In the case of the peripheral mechanism, if cortisol levels, the stress hormone, are high, persistently high and dysregulated, these are the conditions which favor a reprogramming of your immune monocytes, and in particular, the glucocorticoid receptor, which is in receipt of the cortisol, downregulates. And so what we have are so-called glucocorticoid insensitive inflammatory monocytes. These change their character such that they are attracted to the blood-brain barrier, which is normally an impenetrable barrier. It then becomes leaky, and it's the chemokines which attract these monocytes into the central nervous system. So it's like a pincer movement of a central mechanism and a peripheral mechanism coming together to increase inflammation within the central nervous system. This is stress which is high intensity, and it's essentially uncontrollable. And I'm always reminded of the similarity between the sickness, which is caused by infectious pathogens, the phenotype of which is essentially social withdrawal at a population level. That's adaptive, so that pathogens are not spread to other members of a population. But if we look at other behaviors, which are essentially caused by the release of cytokines, we see malaise, anhedonia, hypersomnia, anorexia, all symptoms associated with depression. I think it's instructive to think that just as stress releases cortisol to cope, for an organism to cope, sickness releases pro-inflammatory cytokines to cope. And there's any amount of evidence out there. This is a relatively recent meta-analysis looking at cytokines in the blood and associations with major depressive disorder. And you can see if you look at interleukin 6 or tissue nephrotic factor alpha, TNF alpha, that there is a positive effect, that these levels of cytokines are raised in those individuals diagnosed with major depressive disorder. And in fact, work in Cambridge by Golem Kandekar in the Department of Psychiatry, with Peter Jones and others, has shown that high serum levels of interleukin 6 C-reactive protein can predict later onsets of depression and psychosis. So there's a quite persuasive evidence out there that cytokine inflammatory reactions caused by stress and other factors can lead to later outcomes, including depression. So how does this all work? So let's imagine now we have a situation where there is uncontrollable stress, particularly early in childhood. Cortisol is released from the adrenal cortex. High levels of cortisol, every cell, virtually every cell in the body expresses the receptor for cortisol. The receptor, the glucocorticoid receptor, is a nuclear receptor. And when bound to cortisol, migrates into the nucleus, where it has strong and potent genomic effects. These epigenetic effects are simply chemical modifications of the genome. They can either, this complex, the glucocorticoid receptor bound to cortisol, can either bind with various transcriptional factors to cause methylation of the DNA, which can silence or activate genes, particularly inflammatory genes, or it can work from a mechanism of demethylation. Alternatively, it could work through modifications of histones. Histones are proteins around which protein is wrapped. And the common modifications are caused by these enzymes, histone acetyltransferase, adding acetyl groups, or the stripping away of acetyl groups. Just remember this enzyme here, HDAC, histone deacetylase. I'll come back to this. But essentially, these epigenetic modifications are very likely to happen early in life. The plasticity for epigenetic modification is high during childhood and infancy. And these effects, these so-called stress memories, are persistent. Looking in humans, this is the celebrated study of Liz Binder, looking at this protein, FKBP5. It's a chaperone protein, the gene which codes a protein, which essentially operates to regulate the HPA axis. Now, cortisol works through a negative feedback system. So the system is always designed to shut itself off. It's not adaptive for an individual to constantly release cortisol and not have a regulatory shut-off valve. FKBP5 is a protein which has been implicated in childhood trauma. We know that demethylation of this gene associates with early childhood trauma. And data which has been published by the Binder group shows that there is a risk allele which amplifies the effect of demethylation, and it inversely relates with these inventories of the intensity of early childhood abuse. The point about this is that if you have this risk allele and there is childhood trauma, then essentially become insensitive to glucocorticoids. The stress systems goes into overdrive and it simply can't shut down. Okay, so this is an example whereby a risk allele has amplified the effects of early life stress. And in the case of rats, and I'll be talking quite a lot about rats in this talk, this is work from Mike Meany looking at epigenetic modifications and how it can affect maternal behavior. Let's imagine that we have an environment where there are low levels of resources, high levels of predation, there is social turmoil and aggression in a social colony. What that leads to is poor mothering styles. So here in this example here, if we have an environment just like that, we have a mother here which is low licking, low grooming, and is not taking care of the offspring. The epigenetic consequences of that interaction or that lack of interaction is increased methylation of the promoter for this glucocorticoid receptor. NR is the nuclear receptor, this code is the glucocorticoid receptor. This increased methylation means that this growth factor is unable to bind, and so you get reduced expression of the receptor. Now, if there's reduced expression, then there's nothing for cortisol to act on to shut down the stress response. And so what happens in the offspring are high levels of cortisol. In rats, we call it corticosterone. You have high levels of anxiety, and these behavioral traits are carried from one generation to the next as an epigenetic signal. By contrast, mothers which are caring show high licking responses, high grooming responses, show demethylation of the glucocorticoid receptor, and you see increased expression of the receptor, an opposite behavioral trait, i.e. low anxiety. The offspring subsequently show low anxiety, become good mothers, and so on and so forth. I think these are very important demonstrations of the interactions between the maternal care and the offspring and how it can work through the epigenetic modifications as I've described. The procedure which we use in Cambridge to look at early life adversity is the so-called repeated early maternal separation stress. In this procedure, we separate young offspring, normally from the age of postnatal day 2 to 5, every day, right up to postnatal day 20. Normally, the separation procedure is between 3 and 6 hours, and it can happen at random intervals of the day, so you don't necessarily predict when the separation will happen. There's an alternative procedure, a single episode of maternal separation, where the offspring is separated for a relatively short period, but on a single occasion. Sometimes this can have the opposite impacts. In other words, when the offspring are returned to the mother actually shows heightened maternal instincts, better maternal care, and actually the offspring can show later resistance to future stress. In this procedure, this is carried out, as I say, for 20 days. Males and females are included because I've mentioned looking at sexually dimorphic effects of early life stress and later outcomes, including depressive-like phenotypes, are important. But the real question we had of this study is the necessity and sufficiency of the early life adversity for later depressive-like phenotypes, and whether it's necessary for a second stressor to happen in order to express depressive-like or relevant phenotypes. We know already there is ample evidence that this procedure of maternal separation can activate the central immune system. For example, within the hippocampus, we can compare, in serum samples, looking at C-reactive protein, comparing controlled and maternally separated animals, you can see an elevation of the cytokine. If we look directly in the hippocampus, the cytokine interleukin-1 beta is increased. It's increased both at baseline, i.e. post-separation, but this contrast between the controlled animals and separated animals can be greatly amplified with a second stressor. In this case, it's the injection of LPS, lipopolysaccharide, which stimulates the immune systems. So there's evidence here that this early stress accentuates the effects of a later stress on the release of cytokines. And other work looking at behavior shows that early life stress can have quite dramatic effects on reward-related behaviors, which look like anhedonia. Work here by Keith Matthews. In fact, when I first came to Cambridge in 1996, this was the first project I joined. Keith was working on this procedure as a psychiatrist, interested in the effects of stress impacts and adulthood phenotypes, including depression. What he did in this procedure was to put animals into an open chamber and let them explore the chamber while recording their locomotor activity. At the end of the session, he would feed the animals in the same box. And as session by session by session happened, the animals would anticipate being fed in this chamber and their activity levels would increase. This is conditioned activity. What he noted was that animals, either males and females, which had been subjected to early maternal separation, showed a blunting of this conditioned locomotor activity. Other work, for example, by the late Athena Marku and her group, looked at reward thresholds. This is a technique called intracranial self-stimulation. Animals are pressing levers to inject electrical currents into the lateral hypothalamus. These are animals which have been maternally separated or control animals. These are reward thresholds. If reward thresholds drops, that implies increased reward reactions. Thresholds which increase implies anhedonia. And you can see here in our separated animals an increase in this measure of anhedonia, which has been amplified by a social defeat stress procedure. Okay, so it's all very well and good. So looking at these behaviors, in my view, there's two types of behaviors you can measure. There's the so-called fast and dirty tasks. Fast and dirty tasks are very, very easy to implement, but almost impossible sometimes to interpret. My PhD student, Olivia Stupart, has done a meta-analysis. It's not published yet, but she's looked at 3 tasks, so-called fast and dirty tasks of maternal separation stress. This first one here, it's widely used. It's the elevated plus maze. It's looking for a measure of anxiety. Essentially, it's a raised open maze. One pair of the arms is open. Another pair has arms. It's relatively safe, and the animals would prefer those closed arms when the maze is raised off the floor. What you're looking at here is the proportion of time spent in the open arms. If you look actually at the effect size, which are reported over many studies here, the effect sizes are virtually nil, non-existent. So despite the hype that these tasks and anxiety is one of the outcomes, it's certainly not being picked up by this task. This measure of helplessness has been picked up on a task called the forced swim test. Essentially, you put an animal, a rodent, into a beaker of water, it thrashes around, and eventually it gives up in the stress environment. It just becomes helpless to the stress. And what you measure is immobility time. There's very good predictive ability. It can predict antidepressant drug activity, but it's absolutely hopeless at looking at depression itself. And if you look at the effect size it's reporting, they're very disappointing. In fact, really the only sort of positive effect sizes you see are in relatively young adults subjected to maternal separation stress. And finally, anhedonia, which has been picked up here with sucrose preference. You give an animal a choice between water and a solution containing sucrose, and if you show anhedonia, you show a blunted response to the solution laced with sucrose. The measures that we're using here in Cambridge have, in our view, greater translational relevance. These are touchscreen tasks which can be scaled to other species, monkeys and humans. In this task, a probabilistic reversal learning task, a rat is trained to make a binary choice between one stimulus, a conditioned stimulus here, which on a probabilistic basis is associated with food, and on the second stimulus, the probability of getting reward is just 20%. And of course, an animal will learn the discrimination and trying to maximize the reward output on this task. And one of the ideas that we're looking at here is a bias towards the negative feedback. This task provides spurious negative feedback. So if you're responding on the CS+, 20% of the trials will not lead to a rewarded outcome, despite it being the correct stimulus. And you can model all of this with computational approaches. Another measure of looking at depression is so-called fatigue. We can look at progressive ratio schedules. Again, this can be in a touchscreen. An animal needs to make a response to get food, and then you increase the response requirements. So rather than making one response, you make 2. It normally goes up in a Fibonacci sequence. And eventually, just to get one pellet, you are making dozens, sometimes hundreds of responses to get a single reward. And eventually, there will be a break point. The animal will simply give up. And you can measure that break point and understand whether this is something to do with the fatigue or motivation to work on this task. And then the other battery that we include is anhedonia measures such as sucrose preference and conditioned locomotor activity. Okay, this study was, which I'm going to talk about, was funded by GSK. It was a Varsity Award. It's a longitudinal study using maternal separation in rats. The separation procedure occurred sequentially on each day from postnatal day 5 to postnatal day 19. We took blood samples at postnatal day 20 to look at cytokines. We did an MRI scan at this very young age. This is technically quite difficult in these very, very small animals. Further blood collections at P54, anxiety tests, progressive ratio motivational tasks, another MRI task, young adulthood. We looked at sucrose preference, probabilistic reversal learning. But importantly, in this longitudinal design, we included a second stress. All animals, control animals, and the maternally separated animals received a second stress, as well as another final MRI scan to look at structure and functional connectivity. And this work was carried out by my PhD student, one of my PhD students, Ethan Dusha, who's funded by the Gates Foundation, and a postdoc as well. So first thing we looked at was cytokines. We've measured here 23 pro and anti-inflammatory cytokines. You can pick your favorite here. The boring and rather unexciting news here is that even one day after the final separation stress of these animals, there's no evidence of any robust changes in either pro or anti-inflammatory cytokines. We certainly didn't replicate the earlier finding I've shown you on the interleukin-1b. So in our hands, this is not a particularly robust change. That if there are changes in cytokines, then it may be quite transient. It's certainly not a persistent change that you see. However, what we did next was to use bulk RNA sequencing of blood samples and of samples of the prefrontal cortex and the hippocampus. And this is where the story for us became much more interesting. Remember, this is post-mortem. So these animals, these are control animals and maternally separated animals that have had both stresses. One group had early maternal separation stress, but all animals have had a second uncontrollable stress. One gene of all of the genes we looked at in this bulk RNA sequencing stood out. It's histone deacetylase 3. There are 3 isoforms which we've been looking at, but this first isoform is differentially upregulated in the prefrontal cortex, the hippocampus, and in the blood samples. It's of particular interest because we look at the relationship between this differentially expressed gene, both in the blood, prefrontal cortex, hippocampus. There's a strong linear relationship suggesting that there are global effects on this isoform of HDAC3. For me, the story has greater plausibility when we look at this additional gene called ACSS2. It's not a household name necessarily, but it's also upregulated, both in males and in females, which have been exposed to the early maternal separation stress. What is this gene? Well, this gene actually is involved in the utilization of acetate from fatty acids. So it's responsible for the sequence whereby genes, the histone is deacetylated and subsequently acetylated. So it's following this. So there is almost a biochemical validity to these 2 genes which are being differentially upregulated. Some of you who may be following this story about epigenetic modifications in early life stress may have picked up this paper in Nature published just a couple of years ago, pointing out that the engagement of HDAC3 is almost a master gene, regulatory gene in the inflammatory responses to stress. What we're doing now in collaboration with NIH, the student Rachel Smith from psychiatry working with Ed and Bullmore and Petrovertes, we're now looking at single cell RNA sequencing to look at whether this signal is coming from a particular cellular phenotype, glia, neuronal, glutamate, GABA, and so on. The exciting news here is that this gene is particularly there are drugs, not necessarily selective at this point, which we can use to inhibit this gene and potentially rescue any behavioural phenotypes which result. So behaviourally, what did we see with early maternal separation stress? Well, it's all a little bit negative at this point. So we looked at these fast and dirty tasks, elevated plus maze. These are individual data points for the control of maternally separated animals. Absolutely nothing of any interest there. Were the animals with the early maternal separation stress in any way different in their preference from novelty? This chamber here one side of the chamber is novel, one is familiar, the animal's been habituated to it, and you have a choice, so particular preference, again, no preference for either of these chambers. And then just looking at a chamber which is novel, how do they react in the environment? Again, no differences in these animals. So none of these tests, obviously the wrong tests, or they're simply not sensitive enough to pick up any of these changes. We're also a little bit disappointed to learn that with sucrose preference, that actually, again, no difference for this early maternal separation stress in any hedonic or anhedonic responses. The procedure's working, the animals are preferring the higher concentration of sucrose, but there's no interaction with the stress group. However, something did emerge, which subsequently has become particularly interesting to my group, and this is on the probabilistic reversal learning task. We're still looking at this data. There's a lot to do. I don't have time to talk about whether the animals learn from biasing their learning to the negative, non-rewarded trials, or showing blunted responses to the positive outcomes. But what I can tell you is that when animals are placed in this task, their latencies to respond are dramatically increased compared to control animals. So the dark line here are maternally separated animals. This is the control group. And what was curious to us, in all the tasks prior to that, response latencies were no different to control groups. It's only when you place them in this task, which is uncertain and carrying ambiguous information about which of the stimuli are correct, that this impairment or deficit reveals itself. And what was, again, very intriguing, that when the animals were subjected to a second stress, the deficit reappeared compared to the control groups. It's not a motivational deficit or anything, like simply are not moving fast enough. If we look at the latencies to collect food, these were no different between the control and the stress groups. This is something about a cost, a decisional cost in an environment where there's uncertainty. And this is something which, again, has become very relevant in those working in depression, the endophenotype of ambiguity. People with major depressive disorder are intolerant of uncertainty. And uncertainty is a driver which modulates the rate of learning. And what we're looking at now is how learning is impacted by uncertainty using computational approaches. And Becky Lawson, if you're on this call, know that there's a paper that's just come out in Current Biology, picking up the importance of serotonin, perhaps as the signal which is modulating the effects of uncertainty on the rate of learning, so-called meta-learning. Finally, I want to talk briefly about the brain substrates of maternal separation. This is a plug for our imaging centre at West Cambridge. It has a 9.4 Tesla MRI scanner. What that can do for us is give us highly resolved images across development. And here you can see these structural images of a 3-week-old rat, and 3-week-old rats are tiny. So it's impressive that we can get this level of detail and contrast. And we scan these animals on 3 occasions, and finally during sort of later adulthood. And this is work in collaboration with Steve Soriak from the Department of PDN. What we've discovered was 2 things which we've discovered with this approach. The first thing we discovered was that early maternal separation had no effect on the volume of any structure in the brain by itself. What was required was a second stressor to reveal volumetric differences. Now, Ethan Lucia here has partitioned the rat brain into 7 regions, which are easily segmented by anatomical boundaries: the amygdala, cingulate cortex, dorsal striatum hippocampus, and limbic cortex, insular nucleus accumbens, and prelimbic cortex. The only structure which showed an enlargement to the second stress was the amygdala, and this was only evident in animals which had early maternal separation stress. And you'll see that this looks like what I showed you earlier with remaining orphans and neglect. And so there is some translatability in these findings. The real, I think, unresolved question here is, what is the nature of this enlargement? Is this increased synapse formation, reduced pruning, increased immune activity, microglia? So that's something now we're chasing. The other discovery we've made, and this is of Kat Zudorf, who's a PhD student in my group, has used a functional connectivity approach to look at the coupling strength of the bold response in frontocortical areas. Now, she's used a seed-based approach here, and she's seeded the amygdala and asked the questions, which areas of the brain are showing the greatest strength of correlation with the bold response in the amygdala? And the three areas which showed greater activations and greater coupling strength compared to non-maternally separated animals, they were the cingulate cortex, the infralimbic cortex, and the insular cortex, shown there. Now, if you look in the literature and look in the human literature, looking at both functional and affective connectivity of these types of data, you'll see there is a hub of networks which are interconnected and show differences in connectivity strength and directionality. And these are mapping broadly onto the areas which we've seen in our maternally separated animals, including the anterior cingulate cortex, the anterior insular cortex, and of course, the amygdala. So I want to leave time for questions. So I'll just conclude with some remarks here about the fact that maternal or disrupting maternal offspring relationship early in life can have lasting effects on neurobehavioral processes. They're silent if you're looking at the wrong things or using the wrong tasks, and they only reveal themselves in our hands if the animals are subsequently stressed. They're presumably mediated by the repeated activation of stress mediators, cortisol, noradrenaline, and so on. And they must be of sufficient magnitude to overcome any stress inoculation effects. So there has to be a balance struck there. The epigenetic modifications of the so-called stress memories are amplified, we think, by risk alleles, neuroinflammatory processes, and in particular in response to secondary stresses. And I view it's the early life stress which sets the conditions for the amplifications for future uncontrolled stresses, and presumably via activations or changes in sensitivity of the HPA axis and amygdala processing, perhaps through hypertrophy. And I need to thank a lot of people. The team are shown here and their images. I won't go through everyone there. Most of them, I'm sure you all know, that most are based in Cambridge. GSK for funding the longitudinal study that I've discussed. Gates Foundation for funding Ethan. The MRC and Wellcome for their funding through the Behavioural and Clinical Neuroscience Institute. So thank you very much. And questions?
Thank you, Jeff, for that wonderful talk. Join us next time when we welcome Dr. Madeline Lancaster. Madeline is a group leader in the MRC Laboratory of Molecular Biology here on the Cambridge Biomedical Campus. Research in the Lancaster Lab is aimed at the study of the human brain development, and they do this by making use of in vitro model systems called cerebral organoids or mini brains. She'll be speaking to us on exploring mechanisms of human brain expression in cerebral organoids. Don't miss it. For more info on the talks covered in this seminar series and all things neuro related in Cambridge, please follow us on Twitter at CamNeuro and follow the links below. See you next time.